The goal of this video is to talk about gradients and specifically how those are relevant to the concepts of diffusion and osmosis. Diffusion is the movement of molecules or ions from an area of high concentration to an area of low concentration, and osmosis is merely the same thing but specific to water. So this is what we're going to detail in the subsequent slides and throughout this video. One thing I want to point out from the get-go: diffusion and osmosis, that is things moving down their gradients, is always a passive process. And what is meant by passive is that no energy is required. It's like a ball rolling downhill, or this vehicle right here, which has no motor, is just rolling downhill, or at least the motor on this vehicle would be turned off. It doesn't require any energy to move from high concentration to low concentration. Okay, so before we get into the specifics of diffusion and osmosis, what I want to do is just take a look at concentration, the concentration of solutes or solvents, because it's really important. When I'm talking in anatomy about diffusion, I don't really stress the importance of concentrations, but within physiology, it's very important to understand the significance of concentration, and that is merely the ratio between solutes and the solvents. In the body, the universal solvent is water, H2O, and solutes are anything suspended in that water. It could be ions, it could be molecules. So sodium is a solute, potassium is a solute, glucose, which is a molecule, is also a solute, urea can be a solute, anything. Plasma proteins, which we'll talk about at length further on down the semester, functions as a solute within solution or within the solvent. To highlight what I'm talking about with concentrations, I'm going to use the example of chocolate milk. Could be Ovaltine, could be Nestle's Quick, it doesn't matter. In this case, the solvent is going to be the milk. That is the fluid, the liquid component. The solute in this case is going to be the chocolate. Now, I generally like to use the example of Ovaltine because Ovaltine has quite large granules, and you can actually see with your naked eye each granule of Ovaltine. It doesn't have to be Ovaltine. It could be Nestle's Quick. It could be Hershey's. But the chocolate is the solute. The glass on the right is darker than the glass on the left because there is more chocolate added to the glass on the right than the glass on the left. Both have the same volume of milk. Let's just pretend it's 10 fluid ounces of milk. The glass on the left has gotten 1 scoop of chocolate. The chocolate, once again, is the solute. The glass on the right has 2 scoops of chocolate. So the glass on the right has a higher concentration of solutes. And the corollary to that is the glass on the left has a higher concentration of milk. So in this example, we have the same volume of the solvent, but the concentration of the solute, in this case, chocolate, is modified by the amount of that solute. 2 scoops on the right, more concentrated than 1 scoop on the left. Okay, so what we're going to look at in this video is we can modify the concentration by changing the volume of the solvent. That is to say, we can have the same quantity of the solutes in 2 different containers, but if the volume of the solvent is different, there's going to be a different concentration. For example, on the left, there's a large glass with a large fluid volume of milk. On the right, there's a shot glass with a very small volume of milk. Small solvent volume on the right, large solvent volume on the left. Let's just say the left has 20 fluid ounces and the right, I don't even know how many fluid ounces are in a shot glass, but let's say 1 fluid ounce. But they both have 1 scoop of chocolate. It's the same quantity of the solute, but different concentrations, because with a large volume of milk on the left and that one scoop, it's going to be a low concentration of that chocolate and a high concentration of water. The right side, on the other hand, very low volume of the solvent, same quantity of solute, there's going to be a high concentration of chocolate in that right shot glass. Now, when we're talking about concentrations, when we say the words higher or lower, it's always referencing a different area, a different region, whether it's one glass versus another glass, inside a cell versus outside the cell. So please keep that in mind. It's critically important that we're always understanding or conceptualizing concentration rather than merely quantity, specifically when we're talking about diffusion and osmosis. Okay, so let's look at this slide right here, which is showing a high concentration of sodium in the middle of this slide. The brackets on each side of sodium are representing concentration. So the way that is read is sodium ion concentration. Once again, that plus sign should be a superscript. In the middle, you can see we have a high concentration of sodium. That's why I have it much larger than the other sodiums distributed throughout this screen. In all four corners, I have lower concentration of sodium than the interior of this screen. So in this case, sodium would move to areas of lower sodium concentration, and that's everywhere but the center. It's going to spread out from an area of high sodium concentration to low sodium concentration. To be clear, entities, whether they're ions or molecules, don't have forethought where they are going to rationalize where they want to go. It's really just ions or molecules bumping off each other until they are equally distributed within a region, space, cell, what have you. Let's look at glucose in this example. So once again, I said diffusion can happen on the ion level or at the molecular level. Glucose, which has a super high concentration in the lower right-hand corner of this screen, is going to move to areas of lower glucose concentration, and that's really everywhere else in this screen. Or if this were the extracellular fluid or within a cell. Once again, diffusion is the passive movement of an ion or molecule from an area of high concentration to an area of low concentration. Okay, so now I want to talk about osmosis. And I have a crying baby here because osmosis can be challenging. But to be very transparent, it's no more challenging than diffusion. And I'm not suggesting diffusion is easy, but if you understand diffusion, osmosis should be fairly easy. Osmosis is the diffusion of water molecules. So look at this, look at some definitions and elaborate on what osmosis is. Here's one definition of osmosis: the movement of water molecules from an area of high molecular water concentration to an area of low molecular water concentration. Now, I'm going to read this without the words water in it. The movement of molecules from an area of high molecular concentration to an area of low molecular concentration. That's the exact same definition I gave for diffusion. So osmosis is nothing more than the diffusion of water molecules, which is the movement of water molecules from an area of high molecular water concentration to an area of low molecular water concentration. Which brings us to this second definition: diffusion of water molecules across a semi-permeable membrane. I've added the semi-permeable membrane. I will highlight what that means in a subsequent slide. But once again, I really believe this is the best definition of osmosis, or if nothing else, the easiest definition of osmosis. It's the diffusion of water molecules. And a third definition we can look at is as follows: the movement of water molecules from an area of low solute concentration to an area of higher solute concentration. Now we're going to look at some images, some examples of this in these subsequent slides here, but I just want to be clear. The water molecules, that is, are still moving from an area of high molecular water concentration to an area of low molecular water concentration. But the concentration of solutes in two different areas are going to impact or dictate the concentration of the water. So if there is, in this case, low solute concentration to high solute concentration, wherever there's a low solute concentration, like that tall glass of chocolate milk we looked at, wherever there's a low solute concentration, there is a high solvent concentration or high molecular water concentration. And the corollary, where there is a higher solute concentration, think of the shot glass of chocolate milk. There's a higher solute concentration and a low solvent concentration or low molecular water concentration. So the movement of water from an area of low solute concentration to an area of higher solute concentration is saying the exact same thing as the movement of water from an area of higher molecular water concentration to an area of low molecular water concentration. I know that's a lot to wrap your head around. I'm rattling these off very quickly, but as you move back and forth between these slides and we look at the subsequent images, I'm hoping it's going to make more sense. This definition lends to a term that students often use, which states solutes suck. That is to say, anywhere there's a higher concentration of solutes, it's going to suck or draw water to that region. And that's absolutely true, but it's just supporting this definition and truthfully the very first definition or all the definitions we've talked about. I prefer students don't use the term solutes suck because I find they'll use the term, but they don't necessarily understand what they are suggesting. They'll say solutes suck. They'll know water goes to a specific region, but they're not understanding why. And the why is very important because things, ions, molecules, move from an area of high concentration to areas of lower concentration. Okay, so let's look at some examples right here. In all these images, we are going to have 20 water molecules on the left side of this pink semi-permeable membrane. And on the right side, we're going to have 10 water molecules. So if we just count the right side just for fun, 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10 water molecules. That's going to be the case in all of these subsequent images. Here you do see the presence of the semi-permeable membrane, and it is going to be permeable in this example only to the water molecule. The orange circles are representing the solute, and the solute can be anything. It could be sodium, it could be calcium, it could be glucose, it could be urea, it could be plasma proteins, it could be insulin. Any entity suspended in solution within the human body is going to function as a solute, or almost any entity. So the semi-permeable membrane is important because it's only allowing water to pass through that membrane. It's not allowing the solutes to pass through the membrane. If it did, it would convolute this example somewhat. And we'll look at examples of that later on in the semester. But for our purposes right now, this semi-permeable membrane and all these images is only permeable to water. And what I mean by permeable, it would allow water to pass through that membrane. So the question we're going to ask in all of these images is what direction, if any, is water going to move? To the right, to the left, or not at all? So what we need to do is to determine the ratio of solutes to the solvent, or you could do the ratio of solvent, in this case, water molecules, the ratio of water molecules to the solute. So on the left side, there are 8 solutes in 20 water molecules of a ratio of 8 to 20. On the right side, there are 4 solutes, 10 water molecules for a ratio of 4 to 10. If we were to break those ratios down again, 8 to 20 is the same thing as 2 to 5. 4 to 10 is the same thing as 2 to 5. So on the left side, 8 to 20, I just divided by 4. On the right side, I divided by the common denominator, which is 2. Out to 2 to 5, 2 solutes to 5 water molecules, which is the same. So there is going to be no net movement of water molecules because the concentration on the left is the same as the concentration on the right. And to be clear, we're talking about water here. There's an equal concentration of water on the left side of this membrane as there is on the right. Okay, moving on to another image here. Same number of water molecules on the left and right as we had in the previous slide. That is 20 water molecules on the left, 10 water molecules on the right. But we can see now that there are equal number of solutes on each side of this membrane. Previously, that's not what we saw. So we need to figure out the ratio of these two areas. 4 to 20 on the left, 4 solutes to 20 water molecules on the left side of the membrane. And on the right, we have 4 to 10, 4 solutes and 10 water molecules. So the right side has a higher solute concentration. And if we remember the term solute suck, that means water is always going to move to an area of higher solute concentration, which in this case is the right. 2 to 5 is greater than 1 to 5. Water molecules are going to move from left to right. Now, to be clear, it might be better to look at this as 5 to 1 versus 5 to 2. 5 to 1 on the left is a higher molecular water concentration than the right side, which is 5 to 2. More solutes is diluting that water concentration. Okay, so let's take a look at this example right here. The ratio is 7 to 20 on the left and 3 to 10 on the right. Which one has a greater concentration? Now, admittedly, these ratios are not as easy to work with, but what I would do in my mind is the ratio on the right, 3 to 10, I would times everything by 2 so we can get it into equivalents that are similar to the left. So that would be 7 to 20 concentration on the left, 7 solutes to 20 water molecules, and 3 to 10 is the same ratio as 6 to 20. So 7 to 20 and 6 to 20, the left side has a higher concentration of solutes, so water is going to move that direction from the right side to the left. That is osmosis. Osmosis moves from an area of high molecular water concentration to lower molecular water concentration, which is impacted by the number of solutes within a specific region. Okay, here's our last example. 4 to 20, 2 to 10. What direction is water going to move? I'm not going to tell you. If you wanted to respond to this video, reply to this video, give me the answer and I'll let you know if you're correct. Okay, so that is diffusion and osmosis, merely the passive movement of ions or molecules down their gradients from an area of high concentration to an area of low concentration.